Hello, and welcome to this Nature Custom Media webcast titled Driving Innovation in Human Neural Models of Development and Disease. My name is Alison Halliday, and I will be your moderator. Today's webcast is sponsored by Maxwell Biosystems. Human neuronal models are transforming how we study the development of the nervous system, disease mechanisms, and therapeutic strategies. But key challenges remain. Generating the right neuronal subtypes in vitro, capturing functionally relevant phenotypes, and linking molecular changes to neuronal physiology and behaviour. Within this evolving landscape, human in vitro systems provide an important foundation for complementary humanised models which help extend discoveries until more complex human model platforms are established. In this webcast, we'll examine how spatial transcriptomics is being used to resolve subcellular RNA localization in neurons, offering new insight into axonal vulnerability and local translational programs implicated in neurodegenerative diseases such as ALS. We will also highlight advances in systematic patterning strategies that broaden the diversity of human neurons generated in vitro, supporting more biologically relevant and subtype-specific models. Together, these presentations will provide an overview of how advances in neuron engineering and molecular analysis are converging to create a more informative in vitro systems for studying brain development and neurological disease. You can access closed captioning for this event by selecting English British at the bottom right corner of the video player. With us in the studio today is Sandrine de Cruz, Head, Laboratory of Neurophysiology and Neurodegenerative Disorders, VIB-KU Leuven Centre for Neuroscience, Belgium. We also have Barbara Treulein, Professor of Quantitative Developmental Biology, ETH, Zurich, Switzerland. We will then move on to a question and answer session with you, the audience. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says, type your question here, and then press send, and we'll do our best to answer them at the end of the session today. And now, over to our speakers.
Thanks, Alison, for the introduction, and good afternoon, maybe good morning or even good evening, depending on where you are in the world. I also really want to thank Maxwell Biosystems and the team at Nature, Spring Nature, for the invitation and this wonderful opportunity to share with you some of our recent efforts towards understanding mechanisms of axonal maintenance and loss in amyotrophic lateral sclerosis, or ALS. So let me just briefly start with a brief introduction on what is ALS. ALS is a fatal neurodegenerative disease for which there is no cure yet for most cases, so no effective treatment for most cases. Typically, symptoms occur in midlife, with life expectancy after diagnosis between 2 to 5 years on average. It's really characterized by the selective degeneration of unique neurons, the motor neurons in the motor cortex and spinal cord, that innervate our muscles and allowing, therefore, our most basic functions like walking and breathing, but those are lost and lead to this fatal paralysis that is a well-known feature of ALS. In recent years, ALS is also recognized to be part of a continuous spectrum with another neurodegenerative disorder, which is also fatal, frontotemporal dementia, FTD, which is actually a very common form of dementia, with also presenting symptoms typically in midlife, and is characterized by the degeneration of other types of neurons, neurons in the frontal cortex and in the hippocampus, that typically lead to personality and behavior changes, as well as language impairments. And so this continuous spectrum between ALS and FTD is recognized because of some clinical overlap, but also genetic overlap. The most common cause being mutations, genetic cause being mutations in the gene C9 open reading frame 72, but also overlap with pathological hallmarks. And one of the most common ones have to do with aggregation of RNA binding proteins, TDP43 and FUS, which typically in a healthy neuron will be found mostly in the nucleus. But in patient material, they mislocalized, aggregated in the cytoplasm. In the context of TDP43, also depleted from the nucleus, linked to the hypothesis that its toxicity is really caused by a gain of toxicity and loss of function. And these features for TDP43 are really pathological hallmarks in nearly all ALS cases, as well as half of FTD cases. And FUS pathology is a hallmark not just of the ALS forms with FUS mutations, but also FTD cases, about 10%. So most of the ALS forms have an unknown etiology, that we call them sporadic, about 90%. But 10% of the cases are known to be inherited or familial. More than 20 genes have been found proposed to be causative. Here I'm illustrating the 4 most common ones. And I'll be talking, and mostly because that's what we study most in the lab, forms that are caused also by mutations in TDP43 and FUS, and the reason being because regardless of the mutations, as I mentioned, their aggregation it's a major hallmark of nearly all ALS cases and half of FTD. But regardless of the etiology, what is a common denominator of all ALS forms have to do with muscle denervation, which is really an early hallmark of all ALS cases, characterized therefore very early by the loss of neuromuscular junctions, these unique contacts between the axons and the muscles, occurring before the death of the motor neuron cell body. These are been observed in patients, but also in multiple different models throughout the world. And interestingly, also what was shown is that efforts that rescue the death of the motor neuron cell body in mice, these were mouse models, was not sufficient to halt the disease, mostly because in those models, neuromuscular junction denervation, axonal degeneration was still happening. And this is really what prompted me many years ago when my lab was in San Diego and now in Belgium, really start focusing on understanding the molecular mechanisms underlying this early loss of neuromuscular junction, as I believe that targeting the neuromuscular junction could really represent a therapeutic strategy to treat ALS. And so to do that, we use in the lab a wide range of models. We use ALS, as we call ALS mouse models, in particular mice that harbor the human mutations in TDP43 and FUS. Some of these mice have been instrumental for us to understand these mechanisms, in particular the humanized mutant FUS mice, which we generated a few years ago when my lab was in San Diego, were instrumental in the co-development of a therapy using antisense oligonucleotides targeting FUS that we developed together with our colleagues and collaborators at Ionis Pharmaceuticals and the well-known clinician Neil Schneider in the US. And there is indeed currently a clinical trial ongoing for ALS patients with FUS mutations. To really pinpoint to these mechanisms underlying axonal degeneration, neuromuscular junction loss, we use a wide range of tools, including microfluidic devices that allow us to culture motor neurons from our mouse models or iPSC-derived, to really enable us to identify compartment-specific mechanisms of toxicity that would occur in the axonal compartment or at the neuromuscular junction, as we can also model these neuromuscular junctions in the dish. In fact, using a 2D functional neuromuscular junction model, we screen for modulators of neuromuscular junction innervation that would reduce the innervation caused by ALS mutations. And this is really a work done for my long-standing collaborator in Canada, University of Montreal, Richard Robitaille, who actually identified himself a repurposed drug that is currently being tested in clinical trial with a proof of concept to target neuromuscular junctions, and I'm really proud to be part of this trial with him. So TDP-43 and FAS, as I mentioned, they're RNA binding proteins. They play fundamental roles in RNA metabolism, protein homeostasis in the soma. These have been widely studied, but it's also emerging that they also play important roles along the axons and at the synapse where they actually localize, most likely through their properties to undergo oligonucleotide separation in stabilizing, localizing mRNAs, axonal transport, but also local translation. And so this is not a surprise that perhaps that dysregulation caused by stress, aging, mutations, and so forth is likely to be detrimental and contribute to the early axonal degeneration in neuromuscular junction loss. In particular, in the lab, we focus several of these aspects with emphasis of local and their role in local axonal translation. And the reason is because a few years ago, Yone Lopez-Eraskin, when she was working with me in San Diego, she actually first found and demonstrated for one of the first evidence that there is local translation in mature adult axons. As you can see here in this transversal section of the sciatic nerve of adult mice, that there is this green signal as a proxy for translation in axons. But importantly, what she demonstrated is that there is a significant reduction and suppression of local translation in the mice harboring two of the ALS-linked mutations causative of ALS in patients. And this suppression of local protein synthesis caused by mutations was really observed and measured before we start seeing symptoms, and in particular, we start seeing axonal degeneration and neuromuscular junction loss, suggesting that this early demise could contribute to those deficits. I want to highlight that this role of FUS with and link with translation is widely corroborated by independent studies throughout different labs. An additional aspect that I want to highlight is that what Yone also could demonstrate is that this suppression of translation was actually accompanied by a significant increased apparent misaccumulation of the mutant FUS protein along the axons here in the longitudinal section of our sciatic nerve in the context of the mutations, and really prompted us to propose and suggest that this suppression of translation is caused by a gain of toxic properties caused by this misaccumulation of FUS. And this is really what we've been focusing in the lab, try to understand how. And for that, to really identify potentially key axonal targets that are regulating axonal maintenance and better identify targets maybe for targeting and improving muscle denervation, axonal loss, we want to understand simply the role of FUS and TDP fundamental function in the compartment-specific manner along these axons at the neuromuscular junction. And I won't have time to talk about these efforts, but then also as a way is as to map the transcriptomes, preserving spatial and temporal information in the low motor circuitry, the most vulnerable in health and disease, as well as local proteomes, and really understanding what are the mRNAs that are present along these mature axons, which of those are locally translated, maybe brought by TDP and FUS, and which of those, of course, are impaired by in the context of FUS and TDP pathology. And this is really the team, the current team that is working on all these efforts in the lab. So today I really want to focus on this aspect and really to try, there was a strategy that we decided to use to understand and try to address this question on how are FUS mutations impairing axonal homeostasis and protein synthesis that I showed you. And so for this, we decided to really take advantage of the different mouse models that we have in the lab and tools that really allow us to identify basically the cells and subcellular compounds that are most vulnerable in ALS, in the lower motor circuitry, the motor neuron cell bodies, as I mentioned, that we can nicely identify and that are lost early on in ALS models, the motor axons that are also lost in the nerve, and then neuromuscular junctions are denervated. And really by combining this way that we have to identify these vulnerable cell types and subcellular compartments by immunostaining together with unbiased spatial transcriptomics. And when I say we, it's really the work led by a talented postdoctoral fellow who was in my lab, Diana Pioli, and who is now already assistant professor at the University of Padova in Italy, who really combined untargeted spatial transcriptomics using a nanostring platform with this high quality immunostaining that allowed her to digitally nicely segment in the spinal cord motor neuron cell bodies, and about 10 to 20 were pulled and profiled done by transcriptomics in comparison with non-motor neurons. And importantly, we really achieved this as a way of having a positive control, since the transcriptomic profiling of motor neuron cell bodies has been identified. But importantly, what was never done before and achieved for the first time by Diana was to be able to segment the motor axons compared to the non-motor axons and obtain a nice signature and profiling of these axons. And so as a way then to really validate with a much higher resolution the newly identified transcripts, we needed another technology, and there we also applied spatial transcriptomics, now multiplexed, that spatial transcriptomics targeted where we have single molecule resolution and lead to unravel cell-specific and even subcomponent-specific disease transcriptomic signatures. And here was just an example to illustrate the incredible resolution that is achieved here with some cell-specific transcript markers, where we can even find motor neurons in the ventral horn that would be more resistant versus more vulnerable. This is achievable not just at the spinal cord, but importantly for us to validate of newly identified axonal targets, also in axons of sciatic nerves, either in a transversal section or in a longitudinal section of a mature mouse, a healthy mouse, where you can see in color transcript of different cell types that were multiplexed together with immunostaining for axons in white. And I'm just pinpoint here to this example to show the remarkable resolution achieved of a transcript that is known to be present enriched in Schwann cells that are nicely here surrounding the axon. So this gives us confidence that this technology would allow us to start as a first way of validating some of the newly identified transcripts. And so what did we find, or what did Diana find in our unbiased transcriptomic analysis of the sciatic nerve axons? Well, the transcripts found to be most abundant are involved in translation and ribosomes. She could validate that and demonstrate these transcripts are truly located intraaxonally here, for some examples here. But what happened in the context of disease and in the context of our mice that have harbor the FOS mutations? Well, we find also that transcripts most affected are also transcripts involved in translation machinery. This was really validated at the protein level, where Diana can show that the longitudinal axons here, longitudinal section of sciatic nerves, those axons are actually decorated by ribosomal components, as you can see, by immunostaining, and that this component of the translation machinery is altered in the context of the FOS mutations. When we looked more carefully at the transcripts that were most affected in the context of the FOS mutations, we too caught our attention because those are part of the same pathway. And this is an amino acid pathway, or the spermidine pathway, as we know. Spermidine is actually the substrate that is required and key to allow with this two-step reaction using these critical enzymes necessary to allow this unique post-translational modification of EIF5A, this translational factor that is key to release stalled ribosomes from polypeptides that are difficult to translate. And this modification is this iposine, and iposinated EIF5A is actually needed for its activity. And so what we found is that the transcripts of EIF5A and UHH were significantly altered. And so Diana decided to verify whether first we could detect EIF5A at the protein level in our axons, and indeed she could detect EIF5A, but actually importantly the active form of EIF5A, that is with this iposine-specific antibody. But these was significantly affected in the context of the mutant FOS mice, where we had a significant reduction of EIF5A and in particular of the active form. And what was remarkable and that this mutant-specific reduction in active form of EF5A was really specific to the axons, because when Diana stained for EF5A, immunostain or hyposine in the spinal cord motor neuron cell bodies, she actually did not find any alterations in those levels. So really, this is really consistent with also our findings from a few years ago, where we found the mutations in fast suppressing local translation really along the axons. So then, to understand better the correlation and the link between this the defective pathway and whether this was really critical for the suppression of local translation caused by the mutations, Diana was teamed up with Tessa Roberesh, a PhD student in my lab, who cultured motor neurons from our mice in those microfluidic devices. And so first, what she could show is that in healthy motor neurons, mouse motor neurons, she could nicely detect local translation, and in particular, actually detect hyposine EIF5A, and that the levels of hyposine or EF5A was significantly reduced in a mutant-dependent manner, and this was really accompanied by the suppression of local translation, as we demonstrated a few years ago. But interestingly, what she did is that now she added spermidine, the substrate that is needed for the active form of EF5A. She added spermidine to the axonal compartment. And when she did that, what she found is that first there is a nice rescue of the mutant fast-dependent reduction of the hyposinated EF5A, fully rescued. And not only did she rescue the levels of hyposinated EF5A by spermidine supplementation in the axonal compartment, but also this was accompanied by a rescue of the protein synthesis defects observed by the mutations. And this had functional implications where spermidine also rescued now the defects in axonal growth caused by the mutations in FUS. Spermidine was also enabled to rescue neuronal activity deficits measured with the Maxwell system that we have available at our center. We had shown that mutations in FUS compromise neuronal activity, and again there was significant improvement by adding spermidine. And finally, some of phenotypes also improved in vivo with spermidine, including in Drosophila. This is a work done in collaboration with the laboratory of Ludo van den Bosch here at our center, where we demonstrated that there's also improvement in the eclosion phenotypes of these flies expressing FUS mutations. But interestingly, also beyond the FUS mutation, we also found an improvement in these phenotypes in flies expressing mutations in TDP43, suggesting that this pathway is likely to be important beyond the FUS forms. And so with this, I've shown you that spatial transcriptomics reveal axon-specific EIF5A hyposination as an important regulator of axonal protein synthesis and demise induced by FUS mutation, most likely beyond. And so I'd like to finish with some very brief take-home messages where I've showed you that we identified disease-specific transcriptomic changes in a compartment-specific manner, so in adult spinal cords, but for the first time in adult sciatic nerves, using spatial transcriptomic approaches, combining unbiased for the new discovery, but then with targeted spatial transcriptomic with single molecule resolution. Really allowed us to validate some of these emerging findings, where we find indeed mRNAs coding components of translation machinery to be the most abundant in mouse mature axons. And actually, these mRNAs involved in translation were actually altered in a mutant-dependent manner, leading us to identify for the first time this axon-specific EI5A dysregulation contributing to local translation defects and phenotypes associated with mutant FUS, and were rescued by spermidine. And this is likely to be also relevant beyond the FUS forms that we are currently testing. Indeed, we are now diving more into the molecular mechanisms to really demonstrate that this compromised EI5A position is critical by manipulating some of the enzymes using genetic approaches in vitro and in vivo, and not just in FUS models, but also in many of some of the other models like TDP or SOD1. And with this, I'd like to end here and really thank the amazing team in my lab, as well as wonderful collaborators, and of course grateful for the funding and funders. And I want to thank you for your attention, and we look forward to our discussion afterwards.
Thank you very much, Sandrine, for a great presentation. I'm very happy to now tell you about our work on programming and profiling human neuronal diversity at single-cell resolution. We are currently in an era where a lot of atlases are being generated, where single-cell genomic tools are used to profile the characteristic of cells in our human body. And so what you can see here is two examples of atlases that have been generated with single-cell transcriptomics to characterize all the cell types that are found in the developing and the adult human brain. And from these atlases, what we learn is which transcription factors are characterizing and are defining the different cell populations, in this case, particularly the neuron types that are found in the human brain. We can also learn how these cell types are being specified during development in differentiation trajectories, with certain transcription factors driving the cell type identity that is being set up, and also how with spatial morphogen patterns, different regions in the brain are specified and are then giving rise to these different neuron types that we find in different regions of the human brain. So what I would want to talk about today is how we believe these human cell atlases can really guide how we can in vitro derive human cell types. And why would we want to do that? Well, if we had access to all these different human neuron types in vitro, we could define different disease models that affect specific neuron types. We could also perform drug screens on these human neuronal cell types. And we could test, for example, toxicity of chemicals for specific neuron types. So what we are really interested in doing is to generate in vitro the large diversity of human neuronal cell types that we can find in the human brain. And so when we started to think about that, we looked into different ways of generating human neurons in vitro. And one very popular method is to overexpress the pioneer neuronal transcription factor neurogenin-2 in human iPS cells, in human-induced pluripotent stem cells. And then over a pretty short time, over just a few weeks, neuronal cells develop, and they are they are looking as neurons. They are also really functional. They fire action potentials, as you can see here. And so this is a very attractive way of generating human neuron types. There is one caveat, and this is based on previous work we had done, where we had sequenced single-cell transcriptomes of these induced neurons made by the overexpression of neurogenin-2. What we could see is, and this you see here in this embedding of the single-cell transcriptomes, is that these neurons that emerge, which you see here on the top right, they are transcriptionally heterogeneous, so there are different populations forming. And when we tried to map these neuron types to these primary atlases, we could see that they have a really confused identity. These neurons are no neurons that you would find in the primary human brain. They are actually having a mixed identity of peripheral and central nervous system cell types. So this is not an ideal model. And what we thought is that this is not so surprising, actually. If we look at how neurogenin-2 is expressed, and this is now looking at the developing mouse, but it is very similar in the human. So what you can see in this in situ hybridization, in black are cells that express neurogenin-2. And what you can see is that this is found expressed in the forebrain, midbrain, and hindbrain, all along the spinal cord in a similar way. So this transcription factor neurogenin-2 is very broadly expressed in development. But what happens during development is that on top of expression of this pioneer neuronal transcription factor neurogenin-2, you have the secretion of morphogens in very spatio-temporally defined manner, such as FGF8, Wnt, retinoic acid, sonic hedgehog, and BMP that have secretion along this anterior, posterior, and dorsoventral axis during nervous system development that further specify regional identity. And so this is exactly what we thought to harness in this project that was driven by Sichuan Lin and Jasper Janssens, 2 postdocs in the lab, that took advantage of the knowledge of these morphogens that drive regionalization of the developing human brain on top of a forced overexpression of neurogenin-2 as a neuronal pioneer factor. And so what they did was to define a screen where iPS cells would be induced for NGN2 and at the same time be exposed to morphogens, anterior-posterior patterning morphogens, as well as dorsoventral patterning morphogens in many different combinations and concentrations, such that 288 different conditions were generated. And at the end of this experiment, every condition was barcoded using combinatorial barcoding and then sequenced with single-cell RNA sequencing. We used here parse bioscience-based single-cell transcriptomics. And the result you can see here. So we obtained a large data set that contained 200,000 cells, roughly, that could be clustered into 101 clusters. What you can see is here that the control cells really were found only on the edge of this UMAP embedding, and the different morphogen conditions that you see here at the bottom were really opening up the space of neuronal identities that we could generate. You can, for example, see how the dorsal versus ventral axis with BMP and sonic hedgehog plus pomorphamin really defined the left and right side of this UMAP, and then these other AP axis generating morphogens were further defining this landscape. So what we can do is we can map the cell types that we obtain in the screen to these primary atlases that I showed you in the beginning. What we could see is that these neuron types reflect neurons of the central nervous system as well as the peripheral nervous system. They reflected neuron types that can be found in the forebrain, midbrain, hindbrain, spinal cord, as well as sympathetic and enteric nervous system, as well as different ganglia. And we had different neuron types from glutamatergic neurons via cholinergic, noradrenergic, as well as some neuroblast cells. So these are less mature cells. Now to visualize how certain morphogens really drove the emergence of different neural types, you can see this in these videos that play, where you can see as increasing concentration of retinoic acid and KIR as a Wnt activator, you can see the different clusters were made. So this really shows how these morphogens opened up the landscape of neuronal types that can be produced. We can show this in a more quantitative way here, where we actually show on the left all the different clusters that can be generated with their different annotations in regions and neurotransmitter types, as well as division. And then along the x-axis, what you can see is increasing concentration of retinoic acid, as well as on top increasing concentration of BMP4. And so what we could observe is that when we purely vary retinoic acid concentration, we initially generate these clusters found here at the bottom. As we increase retinoic acid concentration, we are moving to form different neuronal clusters. And now when we leave out retinoic acid or have it as a very low concentration and increase BMP4 concentration, we could see that we get a whole different set of neuronal clusters that form. Now, when we add BMP4 on top of retinoic acid, we again moved away from the initial set of clusters into yet another area of neuronal clusters. And of course, these different clusters are defined by the expression of different transcription factors that were induced on the basis of the presence of these morphogens together with neurogenin 2. So you can see these neuronal clusters are really distinct with specific transcription factor modules. And what we can do on the basis of this data is actually infer the regulatory network that gets induced by these different morphogens. And you can see this here in this network where the different morphogens that we supply to these developing induced neurons are central, and you can see how they are connected to different transcription factor regulons that get controlled. Some of these get controlled by single morphogens or by the combination of different morphogens. And so this is the transcriptional regulatory landscape that really defines these very distinct neuron types that form. So far, we've been looking at how these patterned eye neurons are distinct on a transcriptional level, but there's one big question that remains is actually, are these neurons functionally distinct? And to answer that question, we teamed up with Andreas Hillemann's group here at ETH, who are experts in high-density microelectrode array technology. And we made use of these HD MIAs that Maxwell Biosystems provides. And so the idea is straightforward for these HD MIAs. You have a very highly packed chip of thousands of tightly packed electrodes. And on these electrodes, you can culture neurons. And the electrodes are then passively recording electrical signal from the neurons that grow on top of them at a very high spatial and temporal resolution. And so this is a great technology that provides a lot of advantages over conventional patch clamping, for example, because you can record many neurons simultaneously, non-invasively over longer periods of time. And so we applied this technology to induce neurons that were grown in different morphogen conditions. And what we can obtain from this type of data is actually threefold. We can get single neuron activity information from the spike footprint. This is when individual neurons fire action potential. There's electrical signal that propagates along the electrode array that gets picked up by multiple nearby electrodes. And so the result is a spatial pattern of voltages that we call spike footprints. So this defines really individual neuron activity. Second, we can also get network activity, which is a measure of how a population of neurons undergo coordinated bursts. And the third information we can get is actually axonal morphology information, because the activity travels along axons. And as you can see now here in this movie, based on this action potential that propagates along the axon, we can trace the axonal tree of a cell. And so we applied this technology to the patterned eye neurons under different conditions. As you can see here, we picked a subset of the 288 conditions. And what we could see is that we can identify, based on the single neuron activity, distinct neuronal clusters. And when we looked at the different morphogen conditions, we could see that the cluster composition of each of these conditions is very distinct. So on the single neuron activity level, the different patterned eye neurons have really distinct composition. We then looked at network activity, and here we looked at the synchronized bursting, and what we could see is that the neurons that were patterned under different morphogen conditions produced very distinct network phenotypes. Some conditions gave rise to networks with regular synchronized bursting, and others showed more asynchronous and sparse activity. And so this, again, shows that these neurons are not just transcriptionally distinct for the specific patterning conditions, but there is also functional, they are also functionally distinct. So now moving to the third type of information we get from the HD-MIA recordings, the morphology, the axonal morphology. And so you can see here that already by eye, the control eye neurons are distinct from BMP4 treated, retinoic acid treated, KIR treated neurons, or also these combinations of KIR, BMP4 and retinoic acid. So it is clear there are distinct axonal morphologies forming, but we can also quantify this. In this case, we applied Scholl analysis, which is a very classical neuroanatomical method to quantify the morphology of these axonal trees. And what you can see is that based on this UMAP embedding of all the Scholl analysis features, that these different morphogen treatments really generate distinct neuronal morphologies. So to summarize, these HD-MIA recordings showed us that on a single neuron activity level, on a network level, as well as on a morphology level, these eye neurons that are grown under different patterning conditions are really distinct. And there's not just this transcriptional distinctness. So with this, we were actually ready to take the system a level further, and we were wondering whether we can further generate distinct neuron types from a different starting population. So what I showed you so far is that we induce neurogenin 2 expression in IPS cells and then provide morphogens that control the anterior-posterior or dorsoventral axis during brain patterning. And what we were wondering now is that what would happen if we change the input population? So what would happen if we don't start from induced pluripotent stem cells, but we actually already start from neural progenitor cells that were pre-patterned? So in this case, we start with a neuroinduction to generate neural progenitor cells, and we would provide the patterning information at that point. So we would, at that point, pattern these cells along the AP and DV axis. So we did a second set of screen where we had these pre-patterned neural progenitor cells and would, with 96 different pre-patterning conditions, then induce neurogenin 2 on top and profile the generated induced neurons at day 10. So first, let's look at these pre-patterned neural progenitor cells. In order to characterize them, we performed a single-cell multi-ohm sequencing experiment, and we could see that the progenitors that we generate are derived from different regions of the brain as well as retina. What we could actually see is that within these different progenitor populations, we have very different regulatory regions opened that have neurogenin 2 binding motifs. So now within this population of progenitors, the chromatin landscape is very different, and NGN2 will see its binding motif in very different gene areas. And so this made us think, what will neurogenin 2 now do if we overexpress it? Will it generate the same neuron types as we saw before, or will it respect these open regions and only bind to those domains and then make very regionally specific neuron types? And so when we now overexpress neurogenin 2, what we can see is that we generate neurons that are preferentially neurons of the region that we had pre-specified in the progenitor state. So actually this is quantified here, where we can see that the regional identity of the progenitor cells and the neurons that result at the end is highly similar. So there's high correlation for this regional identity. And we can again see that the neurons that we generate have very different neurotransmitter identities. And actually these neurons are now distinct from the ones we had generated before. So we can even further increase the diversity of human neurons we can generate in vitro. Finally, we were wondering whether we can actually, instead of using morphogens to control these downstream transcription factor regulants on make specific neuron types, whether we can actually remove the necessity of morphogens and instead just on top of neurogenin 2 upregulate the distinct transcription factors that define the different neuron identities. So in this case, we would go away from a morphogen screening, but rather move towards a dual transcription factor overexpression system. And so we did this. In this case, Benedikt Eisinger, a PhD student in the lab, was really driving this together with Suchuan Lin. And what they did is they generated these dual transcription factor overexpression vectors with lentiviruses, where we can now, on top of neurogenin 2, induce additional transcription factors. And we picked some that came up in our regulatory network analysis. And what you can see here is that these the additional transcription factor together with NGN2 really effectively induce specific neuron types. So you can see that, for example, neurogenin 2 together with LHX9 induced this neuron type down here, which is actually the population of neurons that highly upregulated the LHX9 regulon. So this makes a lot of sense. Similarly, NGN2 together with NeuroD1 generated these neurons here in the center that were indeed having the NeuroD1 regulon highly active in our morphogen screen. And one very interesting observation is actually that what we could see is that the purity of neuronal cultures is higher if we do this dual transcription factor overexpression compared to a morphogen patterning screen. So this potentially indicates to us that this approach of multiple TF overexpression might be very advantageous when one wants a really homogeneous neuronal population. So with this, I'm at the end of my talk, and I want to summarize that that I showed you how we have been able to program really the vast diversity of human neuron types using a combination of transcription factor overexpression and morphogen patterning. We could show that these neuron types are transcriptionally distinct, mapping to different regions of the brain, but they also have very distinct functional properties such as single neuron and network firing, as well as morphology. And finally, we dissected the regulatory networks that really drive the production of these different neuron types. And we could show that we could potentially, with overexpression of multiple transcription factors, circumvent the use of these morphogens. And with this, I'm at the end, and I want to thank the team that did this work, as well as you for your attention.
Thank you very much to Sandrine and Barbara for their presentations. It is now time for the live question and answer session. If you'd like to ask a question, please type it in now where it says type your questions here and then press send. And to get us started, we did have quite a few that have actually come in already. So let's see how far we can get. We've got about 10 minutes, so I'll kick off with the first one. I think this one's for you, Sandrine. Do you have any hypotheses on the mechanism by which spermidine rescues the neuronal network activity in the cortical neurons?
Yeah, thank you, Alison. Yeah, that's a great question. Yes, so one mechanism that we favor also based on our findings is through the modulation of this new pathway that we identified, the EIF5A hypocinated pathway, that we show to be relevant for axon homeostasis. And so this is how we think that by maintaining axonal homeostasis, by restoring or maintaining local translation, then the axonal network is just happier. And we think this is also relevant for the synapses and ultimately neuromuscular junctions, which are lost early on in ALS. And this is our future directions, actually, where we'll be testing those hypotheses now.
Great, thank you. And we've got another question for you as well. Is it possible to treat the mutant mouse models? And if so, would you expect a significant rescue at the level of phenotype or behavior?
Yes, we're actually currently trying that by multiple approaches. One is indeed by dosing spermidine to some of our ALS mouse models that we have. Spermidine is not just the pathway that we think is targeting, so to make sure that that's the pathway, in particular the dysregulated hyposination post-translational modification of EF5A, we're also implementing and testing whether we could use genetic approaches modulating that pathway that would restore this defective pathway in some of our ALS mouse models, indeed.
Great, thank you. Our next question is, how can molecular changes such as RNA localization be linked to electrophysiological function in neurons? Barbara, would you like to answer that one?
Yeah, sure. Well, I guess some of the local translation generally in neurons is linked to the establishment of synapses, and how neuronal networks are formed is really linked to the transport of RNA to really far away locations within a process and the formation of new synapses. So in that sense, this is linked. We didn't really look at that, right? That would be very interesting to look at also in these eye neurons. Yes, when we transcriptomically analyzed them, we dissociated the cells and kind of ignored what is localized in processes. But that would be interesting to look at.
Great, thank you. Our next question is, what is the impact of cell type on organizing neuronal networks?
So that's also for me?
I think yes? Barbara as well, yeah.
Yeah, I mean, well, cell types, neuronal cell types are of course defined in different ways. And one major aspect that defines that is the morphology. And I guess this is also something we showed, where with these different conditions, we create cells with very different morphologies, and the morphology and the arborization of these cells is of course also controlling how much a cell can network with other cells, how far reaching a cell is. And so there are cells with huge axonal trees, and then there are others that are much more local, connecting in a less complex manner. So in that sense, cell type is very much linked to how networks are being formed.
Great, thank you. I think we've got another one for Sandrine here. What are the main limitations of spatial transcriptomics in studying axonal vulnerability in diseases like ALS?
That is an interesting question. Yeah, so I guess tremendous progress has been made with these tools, and in particular in spatial transcriptomics field. Maybe if one wants to really do discovery and unbiased spatial transcriptomics, there we can do that, and that's what we did. The sensitivity and the resolution, maybe this is where I think it's emerging, even improving. But in combination then with targeted spatial transcriptomic platforms, where there it's really quite enabling now, where we start seeing that the resolution is quite good, especially when we looked intra-axonally in the mouse. Ultimately, one would say these are transcripts. So in the context of disease, to bring it back to disease, I think it's quite unique to be able to discover potential altered transcriptomic patterns. But then one wants to validate those at the protein level, and then of course perform functional assays to demonstrate that they're really relevant in the context of disease. So this is a bit how we see the pipeline.
Thank you, Sandrine. we've got a connected but a broader question for you to follow on there. What are the main challenges in the long-term study of neurodegenerative diseases?
I would say ALS, now bringing it back to ALS, maybe remains a complex disease because it's quite heterogeneous. And so I guess this has been a major limitation also then when we to design properly clinical trials, for example. And so, and perhaps also being a central nervous system disease, targeting the livering, specifically in the CNS, is not an easy task. But we're also seeing tremendous progress there, where I think there's still a lot of work to do, but still some technologies are advancing to help us maybe better target the central nervous system, also with patient stratifications and development of biomarkers that are also quite critical for the patient stratification. And in addition to that, that's also one aspect that we believe is important, is targeting through the periphery, like the neuromuscular junctions that I mentioned, where those are lost early on, and those don't need to be blood-brain penetrance if we have molecules that target those neuromuscular junctions. So, yeah.
Thank you. We've got a question that's coming about personalised treatment. How can advances in neuron engineering support personalised treatment of neurological disorders? Sandrine, do you have anything to add there?
For me, yeah, well, it's a bit in line with what I just said, but indeed also taking advantage that we have access, and there's many consortia around the world where we have access to patient-derived lines, iPSC lines, and they're having a battery of functional readouts that allow us to maybe start stratifying by then testing modulators that we may have, or favorite hypothesis pathway across the labs, and determine, okay, which could be effective in which subtype of patients. This is an avenue for more personalized medicine, for sure.
Great, thank you. You identified approximately 100 transcriptomic states, but only approximately 10 electrophysiological phenotypes. How do you interpret this?
Yeah, I guess that's a question for me. Well, so actually, we did not assess the electrophysiological phenotype of all our transcriptomically defined clusters. We first went with the transcriptomic screening because that's a really high throughput assay that we can run. We then picked our favorite conditions and only assessed electrophysiological properties on those. We did it like that. We still ran a lot of samples because we wanted to have at least replicates, if not triplicates, from different batches of neurons. So we still had to do a lot of HDMI recordings. But it would have been a really major effort had we done that on all the different conditions. I should also mention that the 100 clusters we see that are transcriptomically defined did not come one cluster per condition, right? So oftentimes a condition would give rise to several clusters. And so there is some transcriptomic heterogeneity within a given condition. And what we could show actually in the very last point that I made in the talk is that if we instead of using NGN2 and a morphogen condition, we would come in with multiple transcription factors that we overexpress, we would get much more homogeneous populations of cells. And I think this is maybe this is definitely currently our ongoing efforts to try to expand on that and see how we can use multiple transcription factors to really make very precise populations of neurons, and then assessing, of course, those also electrophysiologically. So I can't say how many electrophysiological types are among all the neuronal clusters we saw because we didn't assess it. But I think there are less electrophysiological types than transcriptomic types in this case. But this is an area that needs to be explored further.
Great. Thank you. We've probably got time for one last question. And this one's, I think, for you, Barbara. How functionally similar are in vitro generated neuronal subtypes to those in the human brain?
Yeah, that's a very good question. I mean, this is very hard to assess because what is the ground truth data here that we have? I mean, institutes like the Allen Institute in Seattle are pushing really the frontier of trying to phenotype human neurons also with their electrophysiological properties in vivo, in brain tissue, let's say. But we don't have this ground truth data for all cells, of course, for all neuron types. So we are limited already I mean, we can get this better from mouse cells, of course, but then the question is how well do mouse cells compare to human cells? And for human cells, I mean, we can get explant culture data, but that is, again, not the true signal. So it's very hard to even benchmark what is and have the ground truth of a real human neuron. What we can much better assess compared to the electrophysiological type is the transcriptome, let's say. And also there we are still away from a real human in vivo neuron. So there is some sort of in vitro background that we see in all these eye neurons. And there's also a neurogenin 2 background that all NGN2 induced neurons have. And so this is something we need to get rid of. And yeah, so there's definitely further work that needs to be done. But it's already a great advance to have many different neuron types available for personalized studies, disease models, drug screens, as compared to just this one NGN2 eye neuron that doesn't mimic anything. So I think, yeah, we are still far, but it's still a great progress, I would say.
Great. Thank you so much. That was, I think, a great question to finish on. So we have had a lot of questions come in. So just to let everyone know that we will get back to you offline because that's all the time we have for the questions today. So I'd just like to thank you both, Sandrine and Barbara, for your presentations and for answering our questions today. And I'd like to thank our sponsor, Maxwell Biosystems, and of course you, the audience, for tuning in to this webcast today. Remember, you can access upcoming and on-demand Nature Portfolio webcasts at www.nature.com/natportwebcasts or via the QR code that's on the screen right now. Thank you so much for watching, and I hope you can join us again soon.